Recently on this channel, we have been talking a lot about autoimmunity in ME-CFS, POTS and long COVID. And in particular, we have been focusing on a certain type of autoimmune phenomenon called functional autoantibodies. Now, these functional autoantibodies appear to play a role in these kinds of conditions. Not a causal one, but one that nevertheless contributes secondarily to the symptoms. These functional autoantibodies seem to be capable of disrupting core bodily processes, of sending out a kind of negative signal that can stop the body from doing certain things, for example, perfusing blood adequately into the muscles, something that can therefore create exercise intolerance. So we've been talking about these autoantibodies and the research into them. But in today's video, I'm going to consider objections to the significance of these autoantibodies. Because if you look around on patient forums or patient blogs, sometimes people say, actually, these autoantibodies are no big deal. And sometimes they draw upon research that seems to suggest this. However, I'm going to be talking today about why these autoantibodies probably are a big deal and are important, at least as part of one component of these illnesses. But first, if you are new to this channel, my name is Patrick Usher and I'm an ME-CFS patient, and this is the place where I aim to present accessible explanations of the research into ME-CFS, long COVID and related conditions so that you can be more empowered into understanding these conditions better. It's also a place where I talk about my efforts at healing from these conditions, so if that sounds of interest to you, please do like and subscribe. Now, first of all, just a quick recap. You can of course watch my previous videos, now linked above, about autoimmunity and these conditions. But basically the autoimmunity revolves around something called a functional autoantibody. These functional autoantibodies do not damage the body, as in other conditions which are more obviously autoimmune, like rheumatoid arthritis. Rather, functional autoantibodies in illnesses like ME-CFS and long COVID rather disrupt core bodily processes. They do this by binding to cell receptors, specific cell receptors, to stop their normal function. As I just mentioned, that could involve, for example, limiting blood perfusion into the muscles or into the brain, thereby creating exercise intolerance or brain fog, or it could involve limiting the capacity of the heart to beat. This kind of thing. I've done videos going into far more detail about the sort of symptoms that these functional autoantibodies might create. However, if you look around in the Twitter sphere or in different blogs, there are some patients who argue, and they mount strong arguments, that actually these autoantibodies are no big deal. And they point out things like patients who, before they were ill, tested for these autoantibodies and they came back positive for them, and then they tested for the same autoantibodies after they became ill, and lo and behold, they were also positive for them, but in exactly the same ways as before and after. They also point to examples of studies such as this one, a very recent paper, Detection of G protein coupled receptor autoantibodies. That's the family of autoantibodies that are more specific for illnesses like ME-CFS, POTS and long COVID. Detection of G protein coupled receptor autoantibodies in postural orthostatic tachycardia syndrome using standard methodology by Satish Raj and colleagues. And what this paper actually found is that when you compared the autoantibody levels amongst healthy controls with the autoantibody levels amongst POTS, that there wasn't really any particular difference. And indeed, that according to the kind of cutoff points that had been used in previous research, a lot of the healthy controls would also test positive for the autoantibody. As a result of this, there is a strand in this research paper that makes the point, perfectly reasonably, that testing for these autoantibodies is not reliable as a means of diagnosing POTS. I mean, this makes complete sense. If healthy people have autoantibodies at essentially the same level as the POTS patients, then can we say that there is actually an autoimmune component to POTS that is particularly causative of symptoms or contributes to its pathophysiology? And so sometimes you will see patients drawing on this paper in order to suggest that actually it's a waste of time to test for autoantibodies, that actually the autoantibodies are nothing much to do with these conditions, and really there's nothing to see here, it's all overblown. Now, I don't actually agree with this, and I'm going to give you 3 reasons why, and one of those reasons is from also the paper that we've just looked at, because there was more to that paper than meets the eye. And so now I'm going to present you with 3 objections to this idea that the autoantibodies aren't so important, and that to simply suggest that because a healthy person and a person with ME-CFS, for example, might have the same levels of an autoantibody and both might test positive, as it were, for those autoantibodies, that because both of these people are having similar results, that actually there's nothing particularly important about autoantibodies in ME-CFS and related conditions. So here are the three objections. The first is that the presence of autoantibodies in the blood does not tell us actually about what the autoantibodies are doing in the tissues of someone. Now remember, the whole problem with autoantibodies in these kinds of illnesses is what they are doing to the cell receptors in your tissues, in the heart, in the blood vessels, in the nervous system, in the brain. And we can't actually, using a cell trend test, work out what the autoantibodies are doing in those parts of the body simply from measuring the concentrations in the blood. Now here's the really important point. Just because you have the autoantibodies as tested in the blood does not mean that you are having autoantibodies binding to cell receptors in the tissues. Because autoantibodies can also bind away from a cell receptor. And if an autoantibody binds away from a cell receptor, it is harmless. It doesn't cause any physiological disruptions. It's not binding to a cell receptor and blocking an incoming signal from the body in order to do something. So this is a very important distinction. And I wrote to Professor Klaus Wirth, who works on a lot of these areas, to ask him how he would explain this, and this is what he said. Whether autoantibodies develop pathological properties probably depends on how and where they bind to a target. For instance, the beta-2 adrenergic receptor. The question is whether they just bind in the functional region of the receptor, in vivo, or simply somewhat away from the critical functional structures. There are many types of binding of biological structures. All of this is still speculative. So this is really the first important objection. A healthy person and an ME-CFS patient might both test for even a high level of autoantibodies. But the person who's healthy does not actually have autoantibodies in the tissues binding in any significant way to the cell receptors. Therefore, their body is still working normally. Whereas the person with ME-CFS has the same test result, but their autoantibodies in the tissues, because of something to do with the nature of their illness, which is upregulating a negative immune response, their autoantibodies are binding to cell receptors and are therefore creating a physiological problem. This issue was actually mentioned in the paper by Satish Raj that we've just looked at. But this qualification in that paper is usually not picked up upon by patients who are discussing this issue. So this is what Satish Raj actually said. So in a section on functional assays, he makes the distinction from the top of this extract. Functional assays evaluate the ability of autoantibodies to activate G-protein coupled receptors. So this is the point. Are they actually binding to the cell receptor? Rather than just evaluating for the presence of the autoantibodies, i.e. in the blood. So previous studies have found, this is crucial, previous studies have found that cardiovascular G-protein coupled receptor activity, this is the receptor of the cells in the heart and vascular system, to both alpha-1 adrenergic receptors and beta-1 adrenergic receptors is elevated when exposed to sera from patients with POTS compared with controls. So this is the point. In the current Raj paper, controls and healthy people have high numbers of these alpha-1 and beta-1 autoantibodies, leading us to think no big deal. But when you had a previous paper that actually looked at whether the cell receptors in the heart and vascular system were activated in response to the blood of POTS patients, the answer was yes, showing us that something different was going on. This has raised the question of whether the presence of autoantibodies in POTS can be used as a surrogate measure of altered autoantibody activity. The results of this current study suggest that exclusively measuring autoantibody presence through an ELISA test, the cell trend test, may not be a good surrogate. And so that is the key point. Now, bearing in mind all of this, that actually we know that there's something seemingly usually going on in patients with illnesses like ME-CFS and POTS whereby there is a binding to the cell receptor, I think that it could make sense to still test using a cell trend test, as long as you bear in mind that the cell trend test in and of itself cannot tell you about whether the cell receptor activation is happening. However, it would seem nevertheless to be rather likely. Now let's move on to the second objection to this idea. And that is, we know from a paper by Professor Scheibenbogen that the presence of autoantibodies in the blood of ME-CFS patients correlates with symptom severity. That is to say, the more of the different kinds of autoantibodies a patient had, and the greater the quantity of the autoantibodies, the iller the patients were. There was a direct correlation with symptom severity. And this ties in with the point I've just made. In a way, you can say, as long as you have the understanding that it requires the cell receptor activation, as long as you know that, if you are testing for autoantibodies in ME-CFS and people are coming back with elevated levels, it is quite likely that that patient is also suffering from the cell receptor activation in the tissues. But I don't know if you've noticed, but I have quite a bad head cold still, but I'm ploughing on. And this brings us to the third main objection, which is simply basically that you can't argue with success. What I mean by that is that we know from studies, for example, of immunoadsorption, I've done a whole video about that, it's a blood washing procedure which takes out the autoantibodies from the body in a process that you can learn about in my video. But we know that patients who have that treatment can actually get a lot better. We know that from studies, and we also know it anecdotally. I've gone through some anecdotal examples of patients improving. So you can watch that video as well. It were the case that these autoantibodies were no big deal in an illness like ME-CFS, then why would patients get better? It doesn't make any sense. The fact is that a treatment like immunoadsorption is capable of removing the autoantibodies from the tissues and thereby removing autoantibodies that might be binding to cell receptors. And if it improves functionality, it tells us that something is going on. And in some cases, people improve quite dramatically. Similar to that, we know from the early anecdotal examples of the use of Berlin Cures 007, an infusion, a drug, which aims to neutralize the effects of G protein coupled receptor autoantibodies, that patients getting that treatment who have long COVID had a profound improvement in symptoms. In fact, it's been reported that all 4 of the patients that we know of who so far had just a single infusion of Berlin Cures actually recovered. And I've done a whole video about what we know about Berlin Cures so far as well, which is now linked above. And again, it wouldn't make sense for that to happen unless the autoantibodies were important. Now, ultimately, how can this question be settled once and for all? Well, at the moment, there is a big phase 2 trial of Berlin Cures going on across Europe. And that trial is looking at how that infusion works out for 114 patients with long COVID. If the vast majority of them improve their quality of life or better than that, then truly this should be a final answer to this question, that these autoantibodies are playing an important role in these kinds of illnesses. My own hunch is that that is exactly what we will see. And I think that really the best way, to the best of my knowledge at this time, for us to understand this, that while a healthy person and a person with these kinds of illnesses may indeed test similarly for the presence of autoantibodies in the blood, it is likely that there are other mechanisms within the illness of ME-CFS and related conditions that are causing those autoantibodies to bind to the cell receptors. There's something about its pathophysiology that forces that binding, and that is what creates the pathophysiological effect. That is what creates the symptoms. And so we need to have that kind of understanding. And therefore it also seems likely that if an ME-CFS patient does test positive for the autoantibodies, it's not unreasonable to make a leap to suggest that actually those autoantibodies are also causing mischief in the body due to something about the overall nature of the illness. Okay, so that's it for this video in which we've looked at this controversy, you could say, around autoimmunity and ME-CFS. I hope you found it of help. Please do leave your ideas down below. Maybe you have other perspectives. It's always good to engage with people in the comments. If you want to learn more about me, please head over to patrickusher.com. I've written various books, one on POTS, one on thirst and ME-CFS and long COVID, which is available as a free download, and I also offer a consultation service. In the next video, we will be talking about whether it might be possible, never mind immunoadsorption, never mind Berlin Cures, etc., whether it might be naturally possible to reduce the number of autoantibodies in illnesses like ME-CFS without just paying for these expensive treatments. So I look forward to seeing you in the next video.